I'd first like to thank the Brain and Behaviour Research Foundation and the Selection Committee for awarding me this prize, jointly with Bob Desimone. I first visited Patricia Goldman-Rakic early on in my career when I'd first started working with marmosets. Her pioneering studies into the working memory function of the primate prefrontal cortex were an inspiration to me. She combined psychology not only with neuroanatomy, but also neurophysiology and neurochemistry in order to provide us with a window onto the cortical organization of working memory function. Her multidisciplinary approach has had a major influence on my research path, and I hope that will be reflected in. So I'm going to focus on some work we've been doing recently looking at anxiety and anhedonia, anhedonia being the loss of pleasure. And these are two major symptoms of neuropsychiatric disorders and also of neurodevelopmental disorders such as autism, and can also be found sometimes in neurodegenerative disorders such as Parkinson's disease. Unfortunately, our ability to treat these symptoms is relatively limited. Marked individual differences in sensitivity to anxiolytics is an obstacle to successfully treating anxiety, and anhedonia symptoms appear to be relatively treatment resistant to most current pharmacotherapies. And so we have to first understand why this particular impasse. And core to this is psychiatric classification. There are multiple phases of psychiatric diagnosis. So, for example, two patients can be diagnosed with depression but have no overlapping symptoms. Even if two patients have the same symptom, it's very likely that the psychological and neurobiological causes are not the same, since the clinical symptoms themselves are poorly characterized. And then finally, even when treatments do work, we don't actually know how. So we can't predict which treatment will work most effectively in which patient. And so for all these reasons, we need better stratification of psychiatric disorders, along with the development of more personalized treatment strategies. So how do we tackle this problem? The first is to recognize there are multiple psychological causes to the symptoms of anxiety and anhedonia. Now, these are word maps showing the kinds of words that people use when describing their experience of anxiety or their experience of anhedonia. And there are many words being used, and this, I think, reflects the complexity and the unique experience that anxiety is for individuals. And so because of this, we need, first of all, to have better translational tests in our experimental studies from both animals and humans in order to reflect the complexity of these different symptoms. We also need better translational measures, because in humans, we often just ask them to fill in a questionnaire and tell us how they're feeling, or we may measure their cardiovascular activity when they're experiencing positive or negative emotions. Whereas in animals, and this is primarily in rodents where the majority of work's been done, they actually measure behavior. They measure a freezing response of a rat, for example, to threat, or they measure the consumption of sucrose as a measure of anhedonia. And so, first of all, we definitely need better tests to capture these different psychological causes, but we also have to understand there'll be multiple neurobiological causes too. So this is a brain map of the human brain showing you a variety of different regions which we know are important for processing positive and threat responses. However, although we've learned a lot from studies in rodents with respect to the subcortical systems, and this includes all the deep brain structures, we have very little understanding of the functions of the cortex and how these control our emotions. And yet the cortex is the powerhouse of the brain. It's what gives us the richness of thought and our cognitive abilities, and it shows marked changes in activity in patients with anxiety and depression. And so we need greater focus on cortical function, and we need a better understanding of when treatments do work, what circuits in the brain they're acting upon to have their efficacy. We focus particularly on the prefrontal cortex, which is a part of the brain found right at the front of our brains. It's in the frontal lobe region. And we know when we image patients with depression and anxiety that there is altered activity in these prefrontal regions. And so we want to be able to understand how this altered activity in distinct parts of the prefrontal cortex contributes to the different kinds of symptoms we see in neuropsychiatric disorders. I work and have chosen to work with monkeys, and I work with monkeys because in a primate brain, the cortex dominates the brain. In a rat, for example, only 28% of the brain is given over to the cortex, whereas in humans, 80% of our brain is cortex. And likewise in monkeys, whether they're marmosets, New World monkeys, or rhesus macaques, Old World monkeys, 60 and 72% of the brain is given over to the cortex. So I would argue if you're going to understand cortical function, you need to understand it in a species that's got more obvious cortical similarities to the human. And importantly, the prefrontal cortex is an important structure that we know is going to be relevant to understanding neuropsychiatric disorders. And if you again compare the prefrontal cortex of a human brain and a rodent brain, you don't have to see the detail to see that it looks remarkably different. Whereas if you look at the prefrontal cortex of a primate brain, marmoset or rhesus, the overall organization looks remarkably similar. And this is showing you either a ventral surface view, what we call the orbitofrontal cortex, which is found just above the eyes, or a medial surface view. But whichever way you look at it, they look much similar to monkeys than rats. And so in our monkey studies, we've chosen to work with marmosets because they're relatively small. They only weigh about 400 grams, which is no more really than a big rat. But they are very easy to breed, and it's easy to keep them in spacious conditions that suit their biological and social needs. And the other gap we're wanting to bridge in studying the monkeys in emotion regulation is with regard to the measures. So in our marmosets, we measure both behavior and cardiovascular activity. And to measure cardiovascular activity, we implant in a one-off surgery a small device into the descending aorta, which allows us to measure their cardiovascular activity remotely in a freely moving animal. So once they've recovered from the surgery, they're back in the home cage with their cage mate, but we're able to measure their cardiovascular activity in response to both rewards and in response to threats. And so the first thing we wanted to look at was with respect to cardiovascular control, because we know that there is an intimate relationship between depression and anxiety and heart disease. So people with depression and anxiety are more likely to develop heart disease, and likewise, people who have heart disease are more likely to develop depression and anxiety. And so we wanted to ask whether any of the changes that we see in the prefrontal cortex in patients with these disorders actually are responsible for the cardiovascular dysregulation that sometimes accompany these disorders. And so we can measure a variety of different aspects. We can measure heart rate, we can measure blood pressure, and importantly, we can measure heart rate variability. This is how rapidly our heart rate is able to change to changes in our environment. And so the more flexible our heart rate is, the higher the variability, the better. And it is known that in patients suffering sometimes from anxiety and depression, that their heart rate variability is quite low, which means their heart rate is relatively inflexible to changes in the environment. And that's not so good. And I'm sure many of you know already that this system is controlled by both the sympathetic arm, which when activated produces the fight and flight and the energizing in us, whereas the parasympathetic system, when activated, produces the rest and digest. And so we're able to measure this balance in the marmosets. And so the first thing we did was we wanted to look at what happens if we make selective activations or inactivations of specific regions of the prefrontal cortex in a marmoset, similar to what we know can sometimes occur in patients with anxiety and depression. So to do this, we, in a one-off operation again, we're able to put tiny little metal tubes into the brain of a marmoset that's targeting a particular brain region. There's little plastic caps that just sit on top of the head. The animal goes back into his home cage for the next year or so with his cage mate. But then we're able to temporarily place a drug into that brain region, and it can either reduce activity in that brain region or increase activity. And in so doing that, we wanted to see which of these brain areas may contribute to cardiovascular control. And although this is a complicated slide, the message is very simple. Basically, the only area that seems to have any impact on cardiovascular control in terms of basal resting state is area 25, which is an area here found in what's known as the subcallosal region of the prefrontal cortex, which we know is an area that's overactivated very often in depression. And what we found was if we reduce activity in this area, it increases our heart rate variability, which is good, making it more flexible. Whereas if we overactivate activity in this region, then we produce a reduction in heart rate variability, which means the heart rate is less flexible, and that's not so good. Whereas all the other areas we've looked at, whether it's adjacent areas 14 or 32 or up in the anterior cingulate cortex, or it's down on the orbitofrontal surface, none of these areas, when manipulated, have any impact on our basal cardiovascular activity. So area 25 has privileged access. So we then went on to actually understand how do these areas, if anything, contribute to the complexity of fear and anxiety. And one really nice way of thinking about fear and anxiety is along a continuum, where at one end, this end, on the right, the pink end, threat is there, threat's in your face, you react very quickly, reactively, and you may even panic. Whereas at the far end, the green end, this is the much more distal threat. Threat may not even be in your environment, and you may well be not worrying about anything in particular. And then you have all these different stages in between. So pre-encounter threat would be an example of, for example, walking down a dark alley at night, where there is no explicit threat, but nevertheless you have that uneasy feeling that you may be in a vulnerable location. And so you may be alert to whatever's going on around you and feel slightly more anxious than normal. Whereas in the post-encounter threat, in the yellow here, this is where the threat may be closer to you because, in fact, you might notice someone walking down the alleyway in the opposite direction. You can't see who they are. You don't know if it's a girl, an old lady, a large man, and you don't know whether they're probably friend, but they could be foe. So you have plenty of time, though, to sort of see the situation and decide what you're going to do, whether in fact it's fine, you'll keep going, or you kind of notice where your exit routes might be. And of course, in those more distal threat conditions, you're able to engage your higher order cognitive areas to decide what's the best strategy. So all these different aspects of threat we can measure in the marmoset. But the only one I've got time to tell you about today is one which we call encounter anxiety, which is relatively distal threat, where the marmoset is sitting in his home cage. He's separated from his cage mate up into the top quadrant. His cage mate's sort of below in the other part of the cage. And an unknown human comes in and stares at the marmoset for just 2 minutes. It's very brief. And what you find is they show a range of different behaviors. They spend time either at the front or the back. They may go high because if they're scared, they go high. They may show these species-specific head and body bobs, and also they make these vocalizations. And we can use all these behaviors to decide how anxious the animal really is. So in this next slide, we're going to see an anxious marmoset. And as you can see, as soon as the human came in the room, the marmoset's already gone to the back of the cage. He's very quiet at the moment. He's taking what's going on in. He's starting to make the sort of head and body bobs that they make when they're rather anxious. And he's also making these zk vocalizations. And so this is a relatively anxious marmoset. In contrast, I'm now going to show you a relatively chilled marmoset. So here we can see as soon as the human came in, he's down the front of the cage. He's having a good old look, wondering what the human wants, hoping perhaps he'll give him marshmallow, his favorite treats. But he's obviously relatively chilled. And these are two perfectly normal marmosets, untouched. And this is just their natural levels of anxiety in response to an unknown human coming in. And so we can capitalize on this and look at the effects of what happens when we actually manipulate those prefrontal brain areas. And so we've gone on and done that. And what we found was that if we reduce the activity in 2 regions on the orbitofrontal surface, that's just above the eyes, the orbits, if we reduce activity in either area 11 or 13, we actually heighten their anxiety score. So anxiety scores above 0 are starting to show real anxious behavior. And we see that in both cases, when we inactivate these regions, their anxiety scores go up. But the anxiety scores also go up when, in this case, we actually enhance the excitability of these areas on the medial surface, both areas 25 and 14. And so again, we've got now 4 different brain regions which either when inactivated or overactivated produce heightened anxiety. And I would argue that's rather like 4 different people coming into the see a psychiatrist in a clinic with anxiety. These are 4 groups of marmosets with anxiety, but they've got anxiety for different reasons. We've manipulated 4 different parts of the prefrontal cortex which are not doing the same thing. So we would argue we're starting to actually understand the underlying causes of anxiety, the distinct underlying causes, which will help us stratify the disorder better than we have been up until now. I'm now going to switch and start talking about the other symptom, anhedonia, the loss of pleasure, because this is also as complex as fear and anxiety. You can have many forms of anhedonia. You can have what's known as consummatory anhedonia, which, for example, is where you get pleasure out of consuming, for example, rewards, such as if you happen to like a glass of Merlot. So when you drink that glass of Merlot, it feels good. You feel pleasure out of it. But if you have consummatory anhedonia, then you don't. You lose that pleasure. Or alternatively, you may still get the pleasure of drinking from the glass of Merlot, but if you in the past used to get pleasure perhaps by the afternoon thinking about the glass of Merlot you're going to have at the end of the day, and you get pleasure out of even the thought of it, the anticipation of it, if you have anticipatory anhedonia, you don't. You no longer get any pleasure out of anticipating rewards. But finally, it may be that you still get pleasure out of anticipating rewards. But if you find out you've actually got to go around the corner to get your bottle of Merlot, and it's COVID, you're going to have to end up standing in spatially separate queues, and it's probably going to rain, then suddenly your motivation may be not quite so much there anymore. And that might be what you call motivational anhedonia, where really you're not that bothered about the glass of Merlot. And so, of course, you can measure all this in an animal. You can measure how much they like drinking their favorite drinks or eating their favorite foods. You can measure how excited they get in anticipation of getting access to their favorite goodies. And of course, you can measure how hard they're prepared to work. And so one paradigm we've been using is the Pavlovian conditioning paradigm, where the animal is presented with a tone, such as a high-pitched tone, that predicts the animal that he's going to get access to his favorite box of marshmallows. Or he may be given a tone, such as a low-frequency tone, where he actually gets access to an empty box. And so what happens over time is once the animal's learnt this relationship, if you look at their blood pressure against baseline, you see that when the tone comes on that predicts the box of marshmallows, his cardiovascular response goes up, but it stays flat when it's the tone that predicts the empty food box. And of course, when he gets access to the box of marshmallows, then his blood pressure goes up even more. And so these are the various, what we call anticipatory phases and the consummatory phases. And then, of course, you can measure their behavior. And marmosets get very excited when they know they're going to get access to their favorite box of goodies. And so they show these rather short, sharp head jerks. And so you can measure them and see again, they show them in anticipation of getting access to the box of goodies. And then, of course, you can measure their consummatory response. And you can measure how much they eat, how quickly it takes them to eat. So using this, we focused on one area that I'm going to tell you about, and this is area 25 again, which is this area we know is overactive very often in depressed patients. And I've already shown you how this area has privileged access to our cardiovascular system. And I've also shown you that this area also heightens anxiety on the human intruder test. But what does it do to our anticipatory and motivational pleasures? It actually blunts them. So what we find here is that normal behavioral arousal or blood pressure arousal to a tone that predicts getting access to the box of marshmallow. But when we overactivate area 25 with a particular compound, then that anticipatory arousal has been blunted. And likewise, if they're prepared to work very hard to get access to that box of marshmallow normally, if we overactivate area 25, they're not. And so now we're showing that this one region, when overactivated, can produce anxiety and at the same time produce anhedonia, selective forms of anhedonia. And that may therefore not be surprising that anxiety is very often comorbid with depressive symptoms such as anhedonia. So we went on to ask the question, well, if we overactivate this area, what actually is it doing to the rest of the brain? Because these areas don't work in isolation, of course they work in circuits. And so we're able to image the brains of our marmosets while we overactivate this region. And what we found was that if we overactivate this area here, area 25, in the context of threat, then what we find is it causes increased activity in these downstream structures of the hypothalamus and amygdala, which we know are important for processing negative threatening information. But interestingly, it also reduces activity in these higher order cognitive areas. And so you might say that at the very time when we actually want our higher order cognitive areas to make sense of the environmental threat we're in fact, if we've overactivated this area, it actually seems to turn our cognitive areas off, which may be why we over-respond in that situation. But interestingly, if we overactivate area 25 in our marmosets in a rewarding situation, then a completely different network is engaged, involving different areas of the brain, which seem to be more responsible for causing the blunting of their anticipatory reward behavior and arousal. So this is a lovely setting where we can actually look at antidepressants and anxiolytics and see how they may act upon this system. And so I'm only going to tell you about one, and that's ketamine, which is an experimental antidepressant that's been used in humans who are treatment resistant to various depressive treatments. And ketamine, if given just once, 24 hours before, you find the patients do show a lifting of their mood, and it can last for up to 3 weeks. So we asked the question, if we give ketamine to our animals when we overactivate area 25, do we ameliorate their anxiety and their anhedonia? And so what we found was that if we give ketamine 24 hours before, and then we overactivate area 25 and look at the effects on their anticipatory reward arousal, we find that actually the blunting is abolished. And so the animals show normal autonomic arousal, having been given ketamine 24 hours before. So the ketamine ameliorates their anhedonia, gets rid of it for them. However, it doesn't ameliorate their anxiety. They still show anxiety-related behavior after overactivation of area 25, despite having been given ketamine 24 hours before. So here's an example of where one drug can reverse one aspect, the anhedonia, but not the anxiety. So in summary then, what I'm hoping to start to show you, what we've been trying to do, is to dissect out the distinct causes of anxiety by manipulating distinct regions of the prefrontal cortex, which will help us ultimately to stratify anxiety disorders more effectively. And related to that, if we can start showing where these different antidepressants and anxiolytics work on the network within the brain, then we'll have a better understanding of which treatments we should use for which patients, and which will be efficacious and which will not be. And so finally, it just leaves me to thank all my colleagues that have really contributed to all this work, both current and past members. Ones who are involved in the work I've talked about today, I've already highlighted them on the slides, but to thank them because they've also contributed to me getting this prize. Thank you very much.